the organizers than the teams. Maybe I'm missing something, but it seems to me that we would expect most genes actually not even to have an effect in this analysis. Like most of the matrix should not have a signal in the essentiality screen, right? And using something like the correlation coefficient you use basically makes that the ranks are more important, right? And when the ranks are only interesting in the outlier regions, right? The center basically dominates the signal.
Okay, actually that question was asked in the forum quite heavily, as you can guess. But our answer is that for some of the genes, of course, you would expect knocking down a gene would kill everything. And for some of the genes, they won't have any effect, which means they will have slightly positive numbers or near zero numbers, right? And for some of them, we would have both positive and both negative values for gene essentiality. Actually, these genes are the interesting ones because if we need personalized medicine type of application, we need to know, given genomic characterization, which gene needs to be knocked down, right? And in this case, we picked ranking based correlation rather than Pearson correlation, for example, or some other type of metric, because if you have a set of patients, for example, training patients, and if you know which ones are responding to a therapy or not, okay, and when you are given a new patient, when you are presented with a new patient, you want to know relative to earlier patients, what would be the response? Actually, ranking makes sense if you have training set, and if you have a new example, you want to know what would be the response relative to previous examples. That's why ranking makes sense. We don't want to pick a particular gene that would kill the cell if it is knocked down. We want to estimate. We want an estimate that would be relative to the earlier patients or earlier cell lines. That's why we picked ranking-based correlation.
I think I understand. Thank you. I have a related question. So did you try and do any assessment on just the outlier signals that are killing cells?
Okay, actually we didn't do that. That's a nice suggestion, but the thing is that if you need some kind of biological evaluation after the challenge or after the results, the thing is that we need to be concentrated on the cancer-related genes, oncogenes, whatever. And that's why in sub-challenge 2 and sub-challenge 3, we reduced, instead of using genome-wide experiments, we did a subset only. And these are the cancer-related genes. But your suggestion is that instead of doing genome-wide, we may pick the genes depending on their noise, on gene essential.
Or lack of.
Yeah, exactly. But we picked genome-wide data to be as wide as possible. And we picked ranking-based correlation because of the reason that I explained.
Thank you.
Hi, thank you for your talks. My question is that for the training and set data that you give to the participants, this gene's essentiality score is given, right? So how do you calculate that score, and what is the minimum, maximum of that score for the essentiality?
Actually, gene essentiality scores are distinct for each gene. But more negative means more killing, and more positive means not killing. But if I remember correctly, the scale are between -3 and +3, mostly. But
so you don't have any maximum, so that's what you have?
Yes, yes. Any other questions to the best performing teams?
Okay, I think this is it. Thank you very much, guys. No, it looks.